Hey everyone, welcome back to lab. Today we're going to go over heart anatomy and talk about echocardiograms, how to read them, and talk a little bit about conditions that can be identified using them. I'm going to leave the bulk of the anatomy to the end of the lecture, but I wanted to quickly go over a little bit of anatomy just so the rest of the lecture makes sense. So you can see here the right atrium, the right ventricle, the left atria is this red thing here, and then the left ventricle. So in some situations, like on an exam, you're going to need to know how to figure out what is the left and the right side of the heart. And that can be pretty tricky if you don't know what you're looking for. But a really easy way to tell is by looking at the thickness of these ventricles. So you can see this ventricle wall is thick compared to this ventricle wall. The one that's thicker is always the left ventricle. So you can see this must be the left ventricle, the left atria, and then this thinner one, this must be the right ventricle and the right atria. And so if we think about why that would be, think about the physiology of this. The left is always going to be thicker than the right because while the right side has to just pump blood to the lungs, the left side of the heart has to pump blood all around the rest of the body. That's a huge job, and you need that extra muscle to get the pressure up high enough. So again, without having to be told, you should know that it's the right atria, right ventricle, left atria, left ventricle. Just a little bit more anatomy before we move on. The aorta, the pulmonary trunk right here, the chordae tendineae, which are these tendons here, and then the papillary muscles, which are these muscles here that attach to the chordae tendineae, and then here we have the pulmonary veins. Did you notice anything odd about what I just said? Well, if you're paying attention, you would notice that what I just said is that these red blood vessels right here are pulmonary veins. But typically, if you see something in anatomy that's labeled a red blood vessel, you know that that's an artery, not a vein. Well, why in the heart is this different? I'll explain why the heart is a special case on this next slide. So let's think about the definition of a vein versus an artery. A vein is blood being carried towards the heart, whereas an artery is blood that is being carried away from the heart. So typically, arteries are oxygenated blood and therefore are shown in red, whereas veins are deoxygenated blood and therefore are typically shown in blue. However, the blood coming from the pulmonary veins just came from the lungs to the heart and are therefore oxygenated. The pulmonary arteries, however, are carrying blood that will go to the lungs and are deoxygenated blood. So if we look at this on using both the lungs and the heart, we can see is, okay, so the deoxygenated blood should come up through here into the right atria and then make its way into the right ventricle and then get squeezed out into the lungs through this pulmonary artery. Then they get oxygenated and then they go back through these pulmonary veins and get dumped into the left atria. And then they're going to be pumped down from the left ventricle and pumped out to the rest of the body. So now that we've talked a little bit about veins and arteries and we've gotten that part all straightened out, I want to move and talk about the valves of the heart. These are very, very important for cardiac function. So here we have the pulmonary semilunar valve. And then down here, you can't really see it. It looks very similar to the pulmonary semilunar valve, but it's the aortic semilunar valve. These both prevent backflow into the ventricles. So, this is going to go out and go to the lungs. And after a heartbeat, if this valve weren't here, the blood would simply go back into the ventricles. It would come out of this pulmonary artery and go back into the ventricle. Here, it would come out of the aortic arch and go back into the ventricle. And that would be really problematic because you're trying to pump blood out, but then it just keeps flowing back into the ventricles. So these valves prevent that. Similarly, there is the tricuspid valve and the bicuspid or mitral valve. These both prevent backflow into the atrias. So if you think about first, blood is filling up in the atria, and then it gets put into the ventricle, and then the ventricle squeezes really hard to squeeze the blood out this way. If this valve weren't here, the blood could simply go back into the atria. So these are really important. When the ventricle is resting, you don't want backflow from this pulmonary artery going back into the ventricle. And then when the ventricle is pumping, you don't want blood to go from the ventricle back into the atria. So this valve is really, really important. So it depends on what part of the cardiac cycle you're looking at, but these are all really crucial valves. You all might remember back in 2020 when a Kentucky representative's wife was found dead in her office. The coroner later found that she had died of a mitral valve prolapse. So thinking about what we just talked about, this mitral valve, it separates the left ventricle from the left atria, and its job is to basically close up so that blood from this left ventricle does not just go back up into the atria, but instead can build up enough pressure to overcome the pressure in the aortic arch and allow for blood to be forced out and circulate around the body. This is your aortic arch, so this needs to build up a lot of pressure, force the blood out, and then around the body. What happened in her case, though, was this mitral valve did not efficiently close up. So instead of having the blood forced out and around the aortic arch, instead what happened was the blood would just run back into the atria. So she could never get an efficient pumping of blood around the body. Instead, it kept just rushing from atria to ventricle to atria to ventricle, and essentially you die of the blood not being efficiently pumped around your body. There are different levels of a mitral valve prolapse. You can have minor ones in which people can live with it for a little while, or in her case it was really severe and the blood was barely moving at all, and it ultimately caused her death, which is really sad. She was only 39. So in just a minute, we're going to start talking about ECGs, but before we get into that, I wanted to talk a little bit about how electrical current moves through the heart, because I think it's going to be really important for understanding what you're seeing in an ECG. So what happens is this sinoatrial node. This is where electrical current in the heart originates. These cells produce an electrical current. The electrical current comes around the right atria, and simultaneously it's coming around the left atria as well. And it sends a signal. Once it gets to this atrioventricular node, a signal goes down the bundle of His and then down these conduction pathways and then branches out around the right bundle branch. So in the last slide, you saw the electrical waves and how they move through the heart. And then this is what it would look like on an electrocardiogram. Well, actually, this is a very nice version of what an electrocardiogram would look like. They're a little messier than this in real life. But what you need to know, what is the most important off this slide, is this P wave, this Q dip, this R, peak, S, and then this T wave. And you'll quite often hear the QRS complex referred to, and that is the Q, R, and S all at the same time. So now if we're going to put all of that information together, if we look here at this ECG, we're going to see this P wave, and that is the electrical current that is going through the atria, both sides, and depolarizing both of those atria, so causing an electrical change. And then this QRS complex, that is the electrical current moving down the bundle of His and around these Purkinje fibers. And then the T wave is those Purkinje fibers, the ventricles repolarizing, okay? One thing that a lot of students want to do is conflate atrial depolarization and atrial contraction. Those are two different things. First, the electrical event has to happen, and then the contraction can happen. Okay, they don't happen simultaneously. So after this P wave, remember the P wave is the atrial depolarization. After that is when you get this atrial contraction. And then you have that QRS complex, which is the ventricle depolarizing. Remember, it moves down that bundle of His, down the Purkinje fibers, and depolarizes the ventricle. After that happens, then the ventricle contracts. And then you have this T wave, which is the ventricle repolarizing. And then after that, you have the ventricular relaxation. Okay? So make sure in quizzes and exams, when you are shown an ECG, check to see, are we asking about a mechanical event or are we asking about an electrical event? Because those are very different. So this video is basically a demonstration of what we would have done if this were an in-person lab. I have put this link up in the lab content under this lab's section. So look under, cardiac anatomy and ECG. ECGs can be super helpful for figuring out different cardiac abnormalities, different electrical problems with the heart. The ones we're going to talk about are cardiac blocks. So a first degree, I don't think you can actually even see this on an ECG. It's very, very mild and doesn't really cause any symptoms. The electrical signals will be a little bit slow, but they all reach the ventricles. The second degree cardiac blocks, however, the block is a little slower and sometimes causes an irregular heart rhythm. And not all of the signals will reach the ventricles. So some of those heartbeats will be dropped. You'll see a P wave, but you won't see a QRS complex or a T wave. The third degree, these are the really serious ones. This is a complete atrioventricular block. So if you remember, when it's going from the atria to the ventricle, that atrioventricular node is blocked. And this is the most severe form. So none of the electrical signal reaches the ventricles. A natural backup system in the ventricles takes over, but the heart rhythm is going to be a little slower and more irregular than normal. So just quickly going to talk about blocks and kind of what they would look like. So if you look at this particular block, you can see that here these look pretty normal. Here's your P-QRS complex. So the P-QRS complex, the T wave, another P-QRS, T wave, P, and then kind of a lag, and then QRS, and a little bit of a lag, and then a T wave, and then a P wave, but no QRS, and then another P wave, and a regular heartbeat. So which kind of block would you think that this is? So we said that this one you're probably really not going to see much on an ECG. In a second degree block, not all the signals are going to reach and some heartbeats are dropped. That's kind of what this looks like. or you have this P wave, and then you don't have any QRS complex or a T wave. So this is a second degree heart block. Okay, so just a quick concept check. Let's think about this question together. After a fairly severe heart attack, the EKG reveals normal sinus rhythm, but only for the P wave. The QRS and the T waves are no longer in synchrony with the P wave. So what is this problem? Well, it's probably the electrical signal going from the atria to the ventricle is blocked because we're getting a normal P wave, but then this QRS and T wave, they're slower. They're no longer in synchrony with the P wave. So what part of the heart could be damaged? Well, going from the atria to the ventricle, that's the atrioventricular node. So it's likely that that's damaged. So if we're not getting any signal through this atrioventricular node, how are we getting any signal through the QRS complex and the T waves? Like how are these electrical signals being made if they're no longer coming through the atrioventricular node? The answer is that the bundle of His takes over and it will slowly send signals down to the ventricles to contract, so depolarizing the ventricles. So these are going to be slower and not in synchrony because they're not being controlled by the same thing as this P wave is. This P wave is getting its signal from the sinoatrial node, and then the QRS and the T waves, they're getting their electrical signals through the bundle of His. So another really important thing that I want you to remember, this is going to be super helpful on exams when you're looking at, say, the pig heart and you can't tell what is what. It's really helpful to be able to figure out what's a vein and what's an artery. Okay, so how do we figure that out? Well, if it's entering the heart, it'll empty into the atria. If it's leaving the heart, it's going to leave through the ventricle. So you can figure out what is what just based off of where it's going in or where it's going out. And here's just some anatomy of the outside of the heart. Look over these next few slides by yourself and make sure you look over the pig heart too. That should be in your lab manual.